understand that this is an interactive meeting to try to be helpful to the families. I understand that Dr. Hirano is with me here, which I very much appreciate. He has a storied history in this area and is a wonderful neurologist, goes without saying. Erin Chown, whom I helped train, and who works for APBD-RF, prepared some slides, which I liked very much, so I will use those slides. May I share the screen?
Yes, go ahead.
Share screen, APBD, share. So, do you see my screen?
Yes.
It looks great. Do you want to go into presenter mode so we can see sort of there? Beautiful.
There.
Looks great.
Okay, great. So Erin organized it very nicely. I guess the apple doesn't fall far from the tree. What you all need to know, if you don't already know, that there's a gene that's in the genome. This is in our DNA, and I hope you see my pointer. And the APBD gene, the gene that causes APBD when it is mutated, is called GBE1. It's one of, let's say, 30,000 genes in the genome. And that one gene is mutated and causes APBD. So the way it works, the gene is like the blueprint. It produces something called a messenger RNA. This is by its name, you can see it's a message that is being sent to a factory where the protein is being made. So again, we have the blueprint here, DNA. It sends a message, if you want, like an order. And in the factory, when they see that request, they make the protein, which is the GBE1 protein, which is pictured here in the middle of the screen. The job of this protein, every protein has a job. What's the job of this protein? This protein does its job by partnering with another protein. This other protein is glycogen synthase. So this other protein, glycogen synthase, puts the glycogen molecules one after the other in a chain. So that's why it's called glycogen synthase. It synthesizes, or it generates, these chains of glycogen. The thing is that if a chain is too long, it will cause a problem. So to avoid the chain from being too long, our favorite protein, GBE, the APBD protein, comes, cuts a piece, and reconnects it upstream, as you see in this picture. And the single chain becomes a fork with two prongs. And then, please use your imagination. These two enzymes now work in tandem. One of them extends the chain, the other one branches from each prong. And if you can imagine now chain elongation, chain branching, and successive occurrences of this, and you then end up with this large molecule of glycogen with many, many branches. This glycogen molecule is the largest molecule in the cytosol, or in the biggest part of the cell. And it's important for it, despite its huge size, to remain soluble and not to precipitate. What happens in APBD is you have a mutation in the blueprint. So when the order is sent, it is sent wrong. And an abnormal mutated form of the protein is made. It doesn't work very well. it cannot branch properly, you get a final product that is defective in the sense that the branches of glycogen are too long. And as a result, this abnormal form of glycogen, which has a name, it's called polyglucosan, instead of staying in solution, precipitates. Precipitates means it kind of comes out of solution, and this abnormal glycogen accumulates over time, forming these big polyglucosan bodies, which then lead to inflammation in the brain and neurodegeneration and the symptoms of our APBD patients. So we have a very good understanding of what goes on in this disease, which is unusual. In other diseases of the brain, because the brain is such a complex organ, we have much, much less understanding. But for this disease, we have a pretty good understanding of what's going on. So now that we know the root cause, which is the point mutation here, what can we do to remedy this problem? So we know the root cause, and we know the various steps of the disease that end up with the pathogenic, or if you like, the toxic accumulation. What can we do to prevent this? Which level can we work at to prevent this bad outcome? And could we get rid of this toxic product? Yesterday and today, you would have heard various approaches tackling the problem from the root cause all the way to the abnormal toxic compound. So I'm going to start at the root cause, and then I'll take a break and I'll ask Dr. Hirano to talk about the next step, and then he and I will go back and forth as needed. If you have a burning question as we go along, please don't hesitate to ask. Let's make this interactive. So at the root cause, these patients, or the patients, your relatives, your friends, yourselves perhaps, who have this disease, you have normal genes for all the genes. That's why you're otherwise a healthy person. In this one gene, you have a mutation. So this gene is defective. Can we give you a normal copy to replace or make up for the abnormal copy that you happen to carry? Yes, we could. And that's what's called gene therapy. So we can take a normal copy of the gene, then we have to package it in something to deliver it to the brain. The brain is composed of 20 billion neurons, that's one type of brain cells, and just as many, if not more, other cells. We somehow need to get this new copy of the gene into all those cells. There are multiple methods that people are experimenting with, but one of the most advanced methods is to use something that nature already provides to us, and that is a small virus, an inoffensive, innocuous, harmless virus called AAV9. This virus has grown, has evolved with human beings for millions of years, and lives in many people without causing any harm. Kind of like bacteria live on your skin or in your gut. So what we do is we take this virus, we remove its contents, and put inside a normal copy of the gene. And because this virus knows how to move around in the body and deliver its contents to all our cells, we put the virus back into the person, and hope that the virus goes and delivers the cargo, namely the normal copy of the gene, to all the cells. And this actually works. The challenge in gene therapy currently is that we can deliver the new copy to approximately 20% of the brain cells, not 200% of the brain cells. And this is a challenge that many groups are working on to overcome. Nonetheless, it is a promising approach, that could treat at least 20% of the brain and spinal cord as we develop better technologies to deliver to a wider number of cells. So this is what we do at the level of the root cause, at the level of DNA. We do gene therapy, and I explained to you how that works. Can we do something at the message level? Can we say, okay, forget about the root cause, it's mutated, it makes a message that contains the mutation. Can we act on the message, on the order that's going to the factory to make protein? We can, and this is done in a very elegant way by Dr. Hirano's group. It's much more complicated to explain, but I would ask him to please go ahead and explain as best he can. Michio, do you want to take it from here?
Okay. If possible, could I share 2 slides?
Yes, I will unshare myself. Let's see, how do I do that?
Serge, in the bottom of your screen, there should be a little red stop
Got it.
share. Yes.
Thank you.
Okay, thank you. So Dr. Benassian gave a beautiful overview here. And so I'm just focusing on one strategy here. So as Dr. Benassian explained, DNA is the blueprint for all of us, and it contains all the information for all the proteins that make up our body. And it has these different chromosomes, and located here on chromosome 3 is this GBE1 enzyme or gene that is mutated and causes adult polyglucosan body disease. So you can see this is the whole chromosome, and that gene GBE1 is only a tiny bit of that here, but it spans a region 270,000 bases or units in length. But that DNA, as Dr. Benassian described, is transcribed or converted into RNA. And that RNA is very long, same length initially, it's 270,000, but then only the essential exons, right? These segments here, 1, 2, 3, up to 16, are spliced together, and all of this inert stuff, which we call introns, is eliminated. So you end up with a final messenger RNA that's about 1% of the DNA size. It's about 2,800 units in length. So that's how genes are organized. They're organized in the DNA level in these long strands, and then the DNA is converted to RNA, and then RNA has to be spliced together into smaller messenger RNA. And this is just another cartoon version of that, the 16 essential exons with the inert introns in between. So that's the way the DNA and RNA are organized. Now, as Dr. Benassian said, most of the mutations that cause APPD are in these exons. They're point mutations. They're single base changes that alter the protein and render it dysfunctional. It inactivates the enzyme activity. But out of more than 40 different point mutations that cause APPD, only one is actually in the inert intronic region here. This is the normal gene, and this segment here is normally inert. But there's a mutation here buried in this intron between exons 15 and 16 that creates a new deleterious exon, an artificial exon, if you want to call it that. And it disrupts the protein. It actually causes this protein to be abnormal and actually shortened a bit. So that's a problem because there's a mutation here in this normally inert area, and it creates this extra exon, and that's harmful to the protein. So the strategy that my colleague, Dr. Orhan Akben, came up with is to use a custom designed antisense oligonucleotides, or ASOs, that you've heard about yesterday. And it actually blocks this region where this intronic mutation is, and it prevents this exon from being incorporated in the final messenger RNA. So you're preventing this mutation from creating this artificial exon, or this deleterious exon. So it's blocking. Oops, sorry. It's blocking the mutation at the RNA level. Does that make sense? Did I go too fast? Does anyone want me to explain any parts of that again? But it's okay. Well, it's a very special kind of mutation because it's not in the normal coding or exon regions. It's in the intron, and you can block. The point is you can block this particular type of mutation with a synthetic molecule, ASO molecule. So that's very exciting. It only applies to a small number of patients who have this particular mutation, and that's vulnerable to the ASO strategy. But Dr. Akben has been able to obtain about more than 20 different ASOs that are targeted to this region, and he's identified a handful that are very effective at blocking this mutation. And so he's tested them in cells from individuals who have this mutation in the skin cells, that we take skin biopsies from you sometimes to grow them and study them. And so he was able to prove that a handful of these ASOs will block this mutation and theoretically stop the disease from progressing in theory. And so we are working with this foundation called NLORMS, who will provide the antisense oligos for patients. It's a process that will take 1.5 or 2 years. And we're hopeful that we can test it in a small number of people who have this particular mutation. So it's not a cure. It's to restore the normal gene messenger RNA and make the normal protein, but people already are already affected by the time we administer this therapy. So we're hoping that we can either slow down or stop the progression of the disease. It's not going to be a cure, but it may really alter the course of the disease. And theoretically, if we catch people early in the course of the disease, we'll be able to prevent a lot of damage. But it only applies to the small number of people who have this particular mutation. Okay.
Thank you so much, Dr. Hirano. Someone asked a relevant question. How does one know if you have the appropriate mutation? Do you want to address that as I put up my screen again?
Certainly. Yes. So nowadays, DNA sequencing is the gold standard for diagnosing adult polyglucosan body disease and most genetic diseases. So you have to have your DNA sequence done and show that the mutation is present. Now, routine sequencing may not pick up that mutation because it's buried in a region that's the inert intron that, and so it's often ignored by many sequencing labs. So you have to make sure that the DNA is sequenced in a lab that has a special DNA sequencing test that will detect this particular mutation. There are only a few labs that will test for this.
Thanks again. All right, so, sorry. Okay, so I will continue. Dr. Hirano, if you see a question in the chat that kind of is appropriate to answer as I'm going along, can you maybe just take it or stop me so we can take it? Because I'm not able to see the chat simultaneously.
Okay.
I will continue now, and if you don't mind, you can help me address questions if they are timely, or we can leave them to later.
The next question is timely because the question is there work being done for those without this mutation? And I think you're going to address that because.
Thank you. Yes, so as Dr. Hirano explained, that deep mutation that some of you have can be addressed in this special individualized way. The gene therapy approach that I mentioned early on would apply to any type of mutation. But as I said, the gene therapy approach is not yet to a stage where we can fix all your cells in the brain. The approach that Dr. Hirano described, in theory, in due course, after they complete all the remaining experiments, should fix all the cells of the brain in the select group of patients who have that mutation. Now, let's go on to another approach. You will remember that I said GBE is an enzyme that works always in partnership with another enzyme. That other enzyme, as I said, is called glycogen synthase. Glycogen synthase makes the chains become long, adds glucoses to each other to make the chain longer and longer. If there was no branching enzyme, these chains would just keep on becoming longer and longer without any branching, and we would get a toxic product that's much, much worse than what APBD patients have. So APBD patients have some branching enzyme activity, but it's reduced. So you have an imbalance. You have too much chain elongation and insufficient branching. So there's, if you think about a balance, there's an imbalance, too much elongation, not enough branching. The idea is, can we bring it back to a balance situation? So to bring it back to a balanced situation, we can either increase branching enzyme, now we're balanced, or why not decrease glycogen synthase, decrease the chain elongating enzyme, which will again result in balance. For therapies, always decreasing something is easier than increasing something. This is in general applicable to life. Destroying a city, like in a war, for example, in Europe, is much easier than building a city. But in this case, it can help us. So reducing glycogen synthase will get us to the balance that we're trying to achieve. So many scientists have been working on targeting glycogen synthase, the other enzyme, for reduction to achieve balance. How can you target glycogen synthase for reduction? You can target it at the same levels we talked about. You can target it at the messenger RNA level. You can target it at the protein level. For the messenger RNA level, what you use is something called antisense oligonucleotide. For the protein level, what you use is something called small molecules. So we can go into some more detail in this if you like. Let me just say that using an antisense oligonucleotide to downregulate glycogen synthase, this has progressed quite a bit and is ready to go into clinical trial in the related disease of APBD called Lafora disease. I mentioned yesterday that everything is ready for that study to start, except we need about $10 million to run the clinical trial. The same can be done for APBD. A trial could start in APBD. although less background work has been done in APBD. Alternatively, the study can start in Lafora disease, and then as soon as there's some efficacy results coming, potentially the drug can be used off-label in APBD. So there's various things, and we can discuss, if you'd like, these practical matters. As for the small molecule approach, the small molecule against the protein, that would be the drug, for example, developed by Maze Pharmaceuticals. I think we have a picture here. This lady, Julie Ullman, spoke yesterday. This company, Maze Therapeutics, has a drug, a small molecule, which you can take by mouth, which will target glycogen synthase for downregulation, and it works very well. The only problem with the drug is that it does not cross the blood-brain barrier. So far, it's not really relevant for APBD because APBD is mostly a brain disease. I'm sure, though, that they are working on finding variations of this drug that can cross the blood-brain barrier and be useful for APBD as well. The advantage of small molecules is by definition that they are small, meaning they can dissolve them in water and take them as a pill by mouth. The other one, the antisense oligonucleotides, have to be injected by lumbar puncture into the spinal vertebral area to deliver it. So much less or much more invasive, if you like. But the thing, it so happens that this drug is kind of ready to go into clinical trial, whereas the small molecule is not yet ready because it doesn't cross the blood-brain barrier at this time. So these are important strategies that would help in preventing the formation of these toxic bodies. I will just repeat briefly because there's one other thing to talk about in the end. So you can act at the gene level by replacing the gene. You can act at the mRNA level if you have a specific mutation that Dr. Hirano described. Or you can target the other enzyme, the glycogen synthase, at the mRNA level with an ASO, or at the protein with a small molecule. So
maybe there's one that you and I can answer, a general question. It says that given there are no treatments for APBD at the moment, how do you care for your patients in clinic?
Dr. Hirano, you take care of these patients, please. Go ahead.
Well, yes. So although there's no treatment for the underlying root cause of this disease, there are certainly treatments that are important to manage the symptoms of the disease. So, you know, the urinary symptoms are very troublesome for people. So we often try medications for that to try to alleviate the urinary frequency, but frankly, they don't work very well. So we do have to resort to other interventions that are such as catheterizations or diapers to manage that. And so it's not ideal, but we do work together with urologists on that. As Dr. Benassian pointed out, this is mainly a brain disease. So there's a lot of spasticity, stiffness. And so physical therapy can help with that. It's important to exercise and maintain flexibility of your joints. Otherwise you can develop contractures. You know, many people develop infections, so those have to be managed, especially bladder infections. So there are aspects of the disease that we can treat to make people feel a little bit more comfortable.
Thank you. A few more questions. When do you anticipate getting adequate funding for your clinical trial? Addressed to me. This has been very frustrating because we the company that developed the drug, Ionis, was set to go into clinical trial, but Lafora disease is just as rare as APBD. And I think they said they are overextended at this time and would not be able to do it. So the Lafora community is trying to find another partner to invest the funds it takes to do the clinical trial. There are at least 2 companies that are seriously interested, and there are serious discussions going on. Hopefully these will pan out and we can start sometime this year. So that's all I can say about the timing of that. The two foundations, APBDRF and Lafora Disease Foundation, should be always talking to each other, because all those drugs, not all of them, but most of the drugs that we're talking about apply to both diseases. And there's many things to be done in tandem, such as something called biomarker development, which is key to all of this. So I strongly encourage the APBDRF to continue strong relationships with the Lafora Foundation. Next question. How can we help to speed up the clinical trial for compound A? Honestly, I don't know. I'm not involved. This is a question to ask. Maybe there's someone from the company here if they want to take it. I know Dr. Losos and Kahlon are not here. I'll move to the next question, but if someone from the company is here and wants to speak.
Hi, my name is Shlomi Schneider. I'm the founder of Golden Heart Flower, GHF, that was founded actually to try to bring a cure to my mom that's been diagnosed 2 years ago with APBD. And we licensed the molecule, the compound A, together with compound E and the other molecule that Dr. Kahlon spoke about and Professor Losos spoke about. And we're excited actually that we're trying now to take it into a clinical trial to the FDA based on the preclinical work that was done, and add and close all the other gaps that still exist to take it to the FDA. And hope also that there will be positive results on the compassionate trial and compassionate use that Professor Losos described. And in order to speed up, what we need is to raise funds as early as possible. So anyone that can connect the company to any source of funds is just great help to speed up and try to get in time to stop the disease to people that is just progressing for them.
Thank you very much, Shlomi, and nice to meet you in this way, at least, and your great work in supporting, the translation of this project. I'm told we need to wrap up. There's one question I'd like to take and get help from Dr. Hiranot in the answer. Is the nerve problem at its origin in the brain or at the distant end? My understanding is that those nerves, the main cell body is in the spinal cord, the peripheral nerves. So in a sense, they are in the central nervous system. And so it's kind of part of the brain. Dr. Hirano, do you want to?
Yes, so yes, the nerve problem actually originates in the cell nucleus, or the cell body in the spinal cord. But I think part of the functional problem is in the axons, the projection out of the spinal cord and into all the way to the muscle. I think that the polyglucosan accumulates in these axons and clogs them up. It's like a hairball getting clogged in a pipe, and it blocks movement of material. So I think it's both parts of this cell are affected, both the central part and peripheral part.
Thank you. All right. We're on a break now. Thank you all very much.